within a structural element, and then some decorating components in the beta loop, or the loop region, and the alpha helices being basically formulating very closely in small regions within these particular structures. So again, we see a menagerie of structural-like elements which basically come together. And when we see these are highly stabilized constituents in regards to how certain regions of the anti-parallel or parallel beta sheets with the alpha helices stabilize those. But today we're going to look at how we can get to these very complex structures. And it's not a simple approach as you may think in regards to just putting a structure and putting the right chemical environment. There's going to be a lot of energy requirement to get it into that lowest energy conformer ultimately. But once it's in that conformer, we're going to see it become stable. I have a video on that. We also have this issue of symmetry, which basically makes a lot of our jobs easier in regards to being able to identify this. So this molecule we're going to have a look at very closely in protein function is deoxyhemoglobin. And there is 2 alpha subunits and 2 beta subunits, which make up the whole 44 polypeptide chains. Each unit is made up of an alpha and a beta subunit, basically then forming 1 subunit, 2 subunit, 3, and 4. This one is a tetramer, meaning it's 4 subunits, or a dimer of an alpha-beta protomer. Now, protomers are basically defined as these repeating multi-subunits. So we have repeating subunits being present in this particular component. So where we see symmetry in proteins, it really simplifies the whole process in us naming and classifying these complex structures. And as we look a little bit with hemoglobin and myoglobin, we're going to see elements of comparity, but less complexity in myoglobin versus what we see with hemoglobin, because again, it has a different function in regards to it still carries molecular oxygen, but the amount of molecular oxygen it actually carries is different. So let's look at some definitions in regards to this subunit complexing constituency. We have a multimer, an oligomer, and protomer. We've already defined protomer. Oligomer is when we have a few subunits maybe coming together to form an associated protein structure, or a multimer where we might see very multiple complexes. And in some cases, we might see 30, 40 different protein subunits come together to form a functional protein in this particular instance of obtaining regulation, control, and ultimately for that protein to actually do its job and its work. So symmetry itself, we have 2-fold and 3-fold symmetry. So in these cases, we call them C3 and C2 respectively. So in hemoglobin, we have a number of axes. So basically, hemoglobin is a C2 symmetrical protein because we can draw a line across that and see the symmetry between the two. Though the issue is the 3-dimensional components in a single piece of paper doesn't give us that real clear illustration. But take my word for it, there is actually a very large level of symmetry between these two constituents in regards to maybe being an opposite reflection in regards to its positioning. So you might have a region here looking down, you're going to see it like this, where it looks on top, but really on the side, they're going to be in a different position and dimension. So we've got to figure out some of this by looking at the rotational symmetry. And what we do, we basically see if they're superimposable or they're staggered, or there's an issue of some capacity to rotate around a hypothetical axis. And this is what we do when we look at our 2-fold and our 3-fold symmetry. So again, there's various forms, and we can basically see those as we move through our course. When we look at the dihedral, this is where we start seeing further compounding, now a multi-layer of symmetry being present in regards to D2 and D4 in regards to having multiple subunits and their orientation, whether they're actually posed on top of each other or staggered or basically in an opposite control position because of their hydrophobicity, their ionic interactions. So again, we have these types of dihedral symmetry in more of additional layers being placed upon the structure of the protein. We have this also isohedral symmetry, and this is very interesting in regards to what we see with viruses and virus protein encapsulation of the very fragile messenger RNA. So these particular components of isohedral symmetry are basically a number 20 equilateral triangles, all basically having an interactive surface and face with a neighboring triangle and basically forming a 12-cornered polyhedron. These particular guys have various symmetry, 1, 2, 3, and 5-fold symmetry throughout. And the interesting thing is these particular units, these triangles, are actually the same protein. So basically, they're making these triangular proteins of the same structure and same primary sequence, and then just putting them together to form this capsid, which basically is going to be hopefully resistant to enzymatic degradation, as well as the changes in the chemical environment. And here is a phage, which basically is a transmittal component. We see these mostly bacterial phages, very commonly used in infecting plants in research. And you might have heard of the tobacco mosaic virus, a very similar constituent in regards to having a capsulated head, a tail, and these tail fibers where basically they become the recognition points in attaching to the host cell. And then there has to be a transmission of the genetic material to overtake the host genetic system to then cause replication of all these individual proteins to reassemble this constituent as progeny of the viral particle. So these capsuloids become very important because they offer a strong and rigid structure and basically enables them to contain the genetic information in the viral constituents and allows it to safely be moved within the body and cause infection. So again, very important in thinking about how individual structures come together to form more complex, in this case, the isohedral structure that we see in virus particles. So we've got some terminologies we need to look at. Tertiary, quaternary, fibrous, globular proteins, intermediate filaments, and prosthetic groups. And we'll go more to the prosthetics group as we go through a bit further when we focus on hemoglobin. We've got the bigger function and structure of examples of fibrous globular proteins, and you need to compare and contrast. Disulfide bond shuffle in regards to the PERM issue, and then hydroxyprotein instability role, protein symmetry, myoglobin, hemoglobin, and we'll talk more about this particular guy as time goes on. So what I'm now going to do is look at another lecture in regards to now